each day is we're finding new ideas about how breast cancer starts and then how breast cancer metastasizes, and these are exciting times.
Every Susan G. Komen affiliate across the country contributes 25% of all of their donations annually to research, not only to find a cure for breast cancer, but also to improve screening and treatment and survivorship. One of the most amazing things that we've been able to do is invest in researchers who are on the cutting edge, who are spending their careers pushing the envelope, really doing some exciting new work that can potentially change the way we treat this disease. I'm Suzanne Stone, and this is the More Than Pink podcast. Dr. Adrian Lee, who works at the University of Pittsburgh, is the Pittsburgh Foundation Chair for Precision Medicine. He is a groundbreaking researcher, Komen scholar, who is doing a lot of work around insulin growth factors and their influence on breast cancer. But he's also got a lot of other new ideas, too. In fact, he's got so many that the university has given him his own lab. The Lee Lab is expanding this work into some really exciting things, like nanotechnology agents in the treatment of early-stage breast cancer. When I read your bio online and all of the things that you're doing, I need a dictionary to really try to understand what it all means. Yep. Can you tell us what exactly about breast cancer treatment are you researching?
So, I've been studying breast cancer for many years, probably 20 years now. It's been my passion and was the first thing I trained in. And obviously that's evolved over time, just as, kind of techniques and ideas about breast cancer have evolved. I think my most current interest is manyfold. I have a relatively large laboratory. I think one of the more exciting areas is the genetics of breast cancer, because we can now sequence DNA rapidly and affordably. This is really being able to sequence breast cancer and look at the changes in DNA in breast cancer. This has really given us kind of really new insight into the disease. And I think that's an area that I've really expanded. It evolves into many areas. To sequence DNA involves these new machines. It involves big data and handling large amounts of data. So I've ended up evolving into many areas I wouldn't have imagined I would work in. But this is a pretty exciting time. We're just, each day is we're finding new ideas about how breast cancer starts and then how breast cancer metastasizes, and these are exciting times. And I would say one of the goals is sequencing the DNA and looking at changes in breast cancer to understand it. I would say outside of that, my lab is very interested in many other areas, clinically targeting growth factor receptors in many other areas.
When you started your career, what got you down this path? How did you venture down this research area with so many cancers out there? Why breast cancer?
That's an interesting question. So I actually trained in London. I got my degree and my PhD in London, and I was very interested in biochemistry, and I was very interested in cancer, and I was interested in hormone action, and the breast cancer was just, I guess, like many things in life, a little bit of luck. A position came available that was studying a breast cancer lab, and I was just fascinated by it, and I have an incredible passion for this. Like many researchers, we don't do this to make money. But we have an incredible, passion to do this and to try and beat the disease. And I feel lucky that someone pays me to do something I love to do. I feel like I'm like an artist or something. Yeah, I really like doing research into breast cancer, and someone, as long as I can still get funding, which is not easy nowadays, but then I can do something I really want to do. And obviously I love, that's why I'm a big proponent of working with Komen and stuff. I do a lot of my time is spent speaking to lay audiences, and I like doing that. If someone shows back an interest in what I do for example, a breast cancer patient, for example, that's the most rewarding experience, when they have an interest in what you do. So, I've just thoroughly enjoyed it, and it's, a passion.
So let's get back to the lab. 20 years ago, when you began researching, if you can kind of look back in 20 years and see the kind of progress you've made, what would be, in your opinion, the most impactful things that you've found that have changed breast cancer treatment or diagnoses?
Yeah, so in that time, breast cancer's, kind of the treatment of breast cancer, the diagnosis, or most things have fundamentally changed. We used to do the majority of the surgeries were mastectomies, and now we've moved to lumpectomies and, much better surgeries. The radiation has changed. We now have much more directed radiation therapy than we used to have. The therapies, for me, my lab is very interested and historically has been interested in trying to identify kind of mechanisms of breast cancer so that we can identify new therapies or improve upon therapy. So I'm very interested in the therapeutic aspect, and that's fundamentally changed. So the use of hormone therapy has been transformative, and that's put breast cancer as the leading kind of personalized therapy disease. And so I was early in my career, I was fortunate to work in this area of kind of when the aromatase inhibitors became available, I was working in that area, and that was just a very hot area. Everyone wanted to understand them, and they've really, helped improve, the outcomes for estrogen receptor-positive disease. I think the amazing amount of information we now have about the disease is what's a little bit somewhat overwhelming. So when I started 20 years ago, we used to really think in what we call a reductionist manner, so really reduce things to simplicity so we can study them. Because if it's just too complex, you can't study many variables at once. And so I worked on this kind of single pathway, a single gene, and a single protein, and I tried to understand that. Now we're in an era of big data, of where I can measure thousands and millions of variables at a time, which is good because cancer is really complex, and it will never be solved with just one thing. It's not one thing. It's thousands of things that have changed. But to do that research now, you need more sophisticated tools than we had 20 years ago. We need computational algorithms. I work a lot with physicists. I work a lot with bioengineers. We have a project, for example, where we're doing 3D bioprinting of mammary glands in the lab, which is pretty exciting, with some bioengineers at Carnegie Mellon University. So it's just we have this incredible amount of new tools, like, for instance, miniaturization of tools, new computer-aided tools. We have an incredible amount of background information now. We've had the war on cancer for 40 years, so we've gained an incredible amount of knowledge. And at the moment, I think the biggest thing is the implementation of that. How do we spent many years trying to understand disease. How do we really implement that understanding now? How do we get better drugs? How do we use the drugs better? And that's a challenge. That's a challenge I quite enjoy, this, what I'd say, that end stage of translation. This really, how do we get that into patients? I think that's the final goal, and that's a real problem. That's a hard nut to crack.
Is that part of the hormone therapies that you're talking about, being able to and insulin and those growth factors and being able to study that? Tell us some more about that.
Yeah. So so the first project I worked on, which is now 20 years ago, actually that was funded by Komen. So my first grant I was pretty young actually. For most independent investigators, I was like 27. I got a grant from Komen, and that was in the early days of Komen when they were pretty small. I think it was 1997 or 1998. And I was working on these things called insulin-like growth factors. So those are basically growth factors that look like insulin. And insulin, as you know, is the main hormone that controls your metabolism, so your glucose levels. And So when you eat, glucose goes up. Your body secretes insulin to try and metabolize all that glucose. And we worked on insulin-like growth factors, which are similar to that, but they more tend to regulate growth. And so we showed that those are dysregulated in breast cancer, showed that they are associated with poor outcomes in breast cancer, and then worked a lot with pharmaceutical companies to develop drugs to target them. And so that went from right from the basic working in the lab, working on cell lines in a Petri dish, all the way up to doing trials in patients. Now, it turns out a lot of those trials weren't successful, and that's one of the challenges of science. Yeah, most of our hypotheses turn out to be not true. Yeah, and but we still work on that, and I think what we've learned from that is you can't just expect to give a drug to everyone. So these drugs, when they were tested by the pharmaceutical companies, were basically just given to every breast cancer patient. And really, what happened was a few patients responded, but not enough to show interest to the pharmaceutical companies. But what we do as academics is to kind of drill down and say, well, if 10% responded, that would still be a good number. We can then give it to those 10% just alone. So we're trying to now figure that out at the end of this long journey. Okay, well, how do we use those drugs? Clearly, we know the pathway is important. We have the drugs. How do we really kind of implement them? And there's some new data coming out on that.
That's very exciting progress when we talk about, creating that cocktail of medicine that's specific. So really, that cocktail is beginning with you in the lab and this idea that you have, and you somehow managed to move that from the Petri dish to the patient.
Yeah, so this fits actually with my other role. So I'm now director for the Institute for Precision Medicine here at the University of Pittsburgh, and under that hat, I direct this concept of, giving more precise therapy, to the right patient at the right time. So, if we could really understand the unique kind of aberrations in a woman's breast cancer, we should be able to give drugs directly to those aberrations and give them much better outcomes than just globally targeting everything. And that's a challenge because, every breast cancer is different, and it's going to be a while before we can do that efficiently, I think.
One of the things that I've also read that your lab is looking at is something that you're calling nanotechnology agents. When we talk about treating early stage breast cancer.
Yep.
That's a little science fiction, kind of a little science fiction Star Trek sounding. What is that?
Yeah, so that's a word that started maybe 20 years ago, this idea of nanotechnology, of things in the nanoscale, and all it simply means is things that are super small. And in the nanometer range, for example. And that encompasses many things. The area that I worked on and do some work on currently is using what's called a nanotube. So it's a form of carbon that is a unique form of carbon that actually forms these very tiny tubes. And they're hollow. And what we showed was that you can put these imaging agents in them and that they are superior imaging for breast cancer. And we published on that. And then a number of groups now have basically put attach drugs to them or put drugs in them, so you can deliver payloads or you can attach, say, for instance, you can heat these things up. You can shine light on them and they heat up and they cause thermal ablation of the tumors. And there's a huge amount of work going on in that. Actually, my wife is also a breast cancer researcher, and she's working on a project here with an expert in nanotechnology who's using them for drug delivery. So the nanoparticle gets to the place, which would be the breast cancer, and then only delivers its payload when it's in the environment of the breast cancer, as an example. So you can stop systemic toxicity and just deliver drugs directly.
That is absolutely science fiction and incredible that you're so far along in that process that you can actually talk about it and you've seen success with that.
Yeah.
So just using that as an example, how long are we talking? It's exciting for us and maybe a new breast cancer patient is listening and saying, hey, that's what I want. Let's do that. What are we? Are we 10 years out? 20 years out? 5 years out?
I get asked that a lot of all the research we're doing. I always say I would want it as soon in the clinic as you would. My goal is to make this thing go as fast as possible, but I also want it to be safe, and I also want to make sure it works. And that means it does take a long time, and it's unfortunate. And, most of our, a drug from discovery into the clinic is 10 to 15 years. Some of them are taking 20 years, We would love that to be a year, but the reality is these things, they're hard to do, and they take time, and there's various reasons for doing that. I think one thing, going back to that question you said, well, what's changed in those 20 years, is that what I've noticed in my research is, and I see this from a lot of people, is 20 years ago when I was doing research and most people were doing research, we were doing it on cell lines in petri dishes in the lab. My lab does, and I see a lot of others, we do much more research kind of in real time on patients with patient samples now, because we have better access to samples and we have better techniques to work on these samples. And so much of what we do now is some of the stuff is going to be translated. We do have some stuff that will be translated very fast, within 1 to 2 years. Much of the work we now do is on drugs that we could probably, within a year, put them into patients. So I think it depends a little bit on the thing you're talking about. Some things, like you said, are going to be 10 years out, and I think some of the work we're doing on the genetics is probably going to be usable, and some of it is either being used now or be usable in the next few years.
It certainly sounds like a lot of what you do is really reliant on the work that others are doing as well.
Yes, totally, yes. No, that's interesting in that the word translation means you need something to translate, yeah? So you require basic science. We need to fund basic science, because if we don't have basic science, we can't translate anything. We need to understand, every thing we do in medicine comes from some kind of basic science observation that's helped us translate that. But the other thing that's very different now is this concept of team science, that when I worked 20 years ago, I could really just work on my own. I have my cells in the lab, and I can work on them, and that's it. That's really changed, in that now there is a lot of push to do team science. There's a lot of, respect's not the word, there's a lot of support for team science. And, if you want to get your stuff published in the top journals now, you better be doing team science. So pretty much every project we have has these multidisciplinary teams. Like I said, we work with physicists, bioengineers, a lot with computational scientists. It's a very, as opposed to the past, it's very multidisciplinary nowadays.
One of the things you said earlier, you talked about patient samples. And
yep.
How can a breast cancer patient, because I'm assuming these are coming from people who are current breast cancer patients.
Yes.
So tell me how that process works. As a breast cancer patient, if I were a breast cancer patient, how do I give you a sample?
So this is interesting, and this is one of the things I am very passionate about. It turns out that there are incredible burdens to using patient tissue, regulatory burdens. And a lot of investigators give up. Their burdens are so high, and the energy to start doing it is so high, they give up and they start to work on cell lines in petri dishes, or they work on mouse models, for example. But one of the things I value is the ability to work on tissue, and that's actually one of the reasons I moved to the University of Pittsburgh, because we have an incredibly large clinic and an incredibly large biobank. So we consent pretty much everyone coming through for what's called an excess tissue consent. So anyone with breast cancer gets offered to sign this consent, and I would say probably pretty much everyone signs it. And it just says, hey, we're going to be doing a surgery on you. There's going to be a lot of excess tissue that is not going to be used for diagnosis. Would you mind if we use that for research? You have nothing to lose. We're not going to give you information back because we're doing research, and that could be dangerous. But as long as you don't mind that, we're going to go and do research to help the next generation of breast cancer survivors. And as I said, pretty much everyone signs that, and that has been an incredible resource. If we don't have tissue, we just can't understand the disease. So, for example, one of the major focuses in my lab at the moment is metastatic breast cancer. And one of the problems in metastatic breast cancer is we have very few studies of the disease. We know very little about metastatic breast cancer. We've had very few studies of the actual metastasis, which is ultimately the thing that, unfortunately, women die from, and men. So we invest a lot of time and effort in obtaining specimens, and we value the patients who donate it, and we value the clinicians and the nurses who do the consenting, and we invest a lot of money. This isn't cheap, building a biobank and maintaining the biobank. I actually maintain that one here at the university. So this is time and effort, but I think it's valuable. I think what we've learned from biosamples or from samples from breast cancer is immeasurable. If you just take, for example, the most simplest, the estrogen receptor. So that's the first ever kind of predictive biomarker in any cancer. It allows you to give what's the most successful targeted therapy, hormone therapy, the most successful and least toxic therapy. And that all came from a person, Bill McGuire, who had the foresight 30 years ago to collect these excess tissues and then do measurements on them. So it's incredibly valuable. It's time-consuming. It's difficult. It's costly. You don't get grants to do this, but I think it's invaluable.
When we talk about how much it costs, it's costly not just in materials, I would assume, but in people.
Yeah.
So when you're looking to fund your research lab and the research that you're doing that will ultimately change the way breast cancer looks in the next 10 years, when we say it costs a lot, what is a lot?
Yes, so that is, so like you said, generally like in most businesses, the people are probably the most expensive part of it. And we have a large team of biobankers. So I run the whole of the university biobank. But in breast, we have, I think, a personnel of 4 or 5 full-time employees that do biobanking. And so you could guesstimate from that what salary costs are. I mean, if we look at our total biobanking commitment across the university, it's in the millions per year. That's millions per year. We fund a lot of that through philanthropy, so it's hard to get grant funding for that. The NIH tends to fund the research on the biosamples, not the collection of the biosamples. And you have to understand that, many of these biosamples, they're not valuable until 10 years later. They're not valuable at the time of collection. They're valuable when you know if that patient had a recurrence or didn't, for example. So this is it's like investing for retirement. This is a kind of, you need foresight to do this, and you need to have, So it's costly. We tend to do that through philanthropy. It's hard because we're constantly trying to raise money to do that. We have, for instance, a full-time employee who is in the hospital collecting, just from our patients with advanced breast cancer, where she will go and collect blood from them, collect tissue from them. I think one thing we have that's unique here is we do have this either what's called an organ donation system or a rapid tissue donation system, or called a rapid autopsy system, whereby if patients are at the end stage of life, they can donate their body for research. There aren't many of those in the U.S., and that gives us a really unparalleled view of what was the final problem. We can take that tissue, we perform an autopsy, we collect all the tissues, we can then analyze it, and we can really then understand, what eventually caused the person to pass away. So we can, no one really wants to talk about that, but ultimately, if we're to end that, we need to understand what the final consequences were. So those are really expensive programs because they get activated, any time of the day, and we have a whole team that gets activated, multiple people. But these are the things we need to do to answer the problem.
It definitely sounds like the investment today is something that pays off in the future tenfold.
Yeah, absolutely.
So looking ahead, what is that piece of research, whether it's happening in your lab or another lab, that's really exciting to you, that you think will ultimately change breast cancer treatment?
I think there's two things. One happens in my lab and one happens out of my lab, and I'm pretty open-minded, so I don't only speak about what I work on. But I think outside of my lab, I think everyone's incredibly excited about immunotherapy. This has really taken off in some other cancers, particularly melanoma, lung cancer, for example, where they're getting great responses to the immunotherapy, real extended responses in diseases that have been very hard to treat. Unfortunately, that's been much more challenging in breast cancer, and clearly more research is done to figure out how that's going to be used. But clearly, I think everyone is excited about that, kind of using the body's own immune system to attack cancer. I think what goes on in my lab that I'm most excited about is these things called liquid biopsies, where due to new techniques and new ideas and concepts, we can now take blood from women and men with breast cancer and measure stuff in the blood that's mimicking what's going on in the tumor. So it turns out that the tumor, when it's growing, secretes and loses stuff into the blood, and so we can then pick that up. So rather than having to, say for example, someone has a metastasis in their liver, rather than having to go in and biopsy that metastasis in the liver, which is very invasive and dangerous, with lots of people now doing research to see, well, can we just look at that in the blood? And our studies and other people's studies suggest, and this is early research, but suggest this is a pretty exciting way to monitor what's going on in the tumor without having to go into the person's body, and then also predict which therapy they should receive or identify which therapy they should receive. So there's already some clinical tests available. So when I said to you earlier about how much of the research we do is kind of like at the cutting edge of clinical treatment, I mean, these tests have now become commercialized, and so we're doing the commercial ones at the same time as we're doing the research ones. We do them kind of side by side. So, but I am excited about that. I think that's giving us, the last 2 to 3 years has given us a brand new view of breast cancer, the genetics of breast cancer. And as I said, it's still early days because we've only had this concept for 2 or 3 years, but already it looks exciting. And I think, I have a feeling that that's going to change how we diagnose breast cancer and then how we assign therapy.
Cannot wait to have that at the doctor's office near you. Incredible work. Dr. Lee, thank you so much for what you do. We really appreciate it, and so do the thousands of women out there listening. Dr. Adrian Lee is part researcher, part fundraiser, full-time pioneer. Funding research is clearly key to changing how we treat breast cancer. The sci-fi of today will absolutely become the reality of tomorrow, but only if we invest today. Susan G. Komen is the largest private funder of breast cancer research, $920 million as of the summer of 2017, and counting. We hope you'll join us at KomenAustin.org. Like to do a big shout out to our iHeartMedia studio partners for making each and every podcast happen. Thanks, Mike. You produced and mixed it perfectly this time. Also wanted to thank our interns, Jacqueline and Mallory, for keeping me in line. Susan G. Komen Austin is an affiliate of the Susan G. Komen Breast Cancer Foundation. If you need resources, information, or just have a comment about this or any of our episodes, we hope you'll reach out to us at podcast@KomenAustin.org. We'll talk with you next time. And always, be more than pink.